For this Brainstem lab, we are going to look at all three portions that make up the brainstem using various digital horizontal sections, and we're going to try to relate some of the internal structure of what we're looking at to what we can observe on the external gross brainstem. So we're going to start more inferiorly at the level of the medulla and then work our way superiorly to and through the midbrain. So when we study the medulla, there's four major sections that we're going to look at that's hopefully going to help us gain a better understanding of the underlying nuclei and tracts that are present within the medulla itself. So our first section that we're going to look at is going to be at the level of the pyramidal decussation. So to understand what the pyramids are, we can look at the sagittal section of the brain where we can see numerous fibers that are radiating through the cerebrum, going to and from all the different lobes. And all of these radiating fibers appear to converge in a bundle in the inferior medial portion of each cerebral hemisphere, and that bundle is referred to as the internal capsule. So the internal capsule has a bunch of ascending sensory fibers as well as descending motor fibers. Right now, when talking about the pyramids, we're going to be concerned with these descending motor fibers that originated in the motor cortex, descended through the internal capsule, through the midbrain, through the pons, and to the medulla. So once these corticospinal tracts are actually within the medulla, they form these characteristic bulges on the anterior surface that we can see from an external view, and we refer to these bulges as the pyramids. So we have two pyramids on either side of midline that are separated from one another by this anterior median fissure. Now, at the inferior portion of the medulla, many of these pyramidal or corticospinal fibers are actually going to decussate to the contralateral side, and they're going to travel down through the contralateral spinal cord. So most of these fibers in these corticospinal tracts are going to decussate, but not all. There's some that are just going to remain on that ipsilateral side as they travel down the ipsilateral spinal cord. So when we look at the internal structure of the medulla at this level, we can see some similarities to the spinal cord. So this image on the left is at the spinal cord at the level of the thoracic region, and the image on the right is going to be in the medulla at the level of this pyramidal decussation. So once again we can see some similarities. So in the spinal cord we had these four large humps back here that were making up our dorsal column, and we have those same four humps, dorsal column tracts, in this level of the medulla as well. Now back to the spinal cord, you can see it's got that characteristic butterfly or H-shaped central gray matter. And if we look in the medulla, we can kind of see some remnants of that central gray butterfly appearance as well. So we'd have these posterior horns right here, and then we kind of have some remnants of the anterior ventral horns as well, starting to disappear in this anterior portion because they're broken apart by these large kind of V-shaped masses. And those V-shaped masses are actually fibers that are decussating. And so this is the actual decussation of the pyramids, or decussation of these corticospinal tracts. Okay, so once again we had these corticospinal tracts that were descending until they got to this level, and many of those fibers are going to decussate and travel posterior laterally until they get to this region. And once they're in this region, they're just going to be diving deep and traveling to that contralateral spinal cord. And they're going to remain roughly in this position all the way down through that contralateral spinal cord. Now those fibers that started off in this location that are not decussating are just going to remain in this location, in this anterior portion, throughout the spinal cord. So when we look at the spinal cord, those tracts that were decussating are forming the lateral corticospinal tracts that are going to be in this lateral white column of the spinal cord. And then those fibers that did not decussate, but they're remaining ipsilateral, are going to be in this anterior white column, and they're going to be the anterior corticospinal tracts. So moving back to our medulla, once again, we can see these dorsal column tracts as they're ascending from the spinal cord. So more medial, we have the fasciculus gracilis that's carrying proprioceptive information, vibration, fine touch from the lower extremity. And then more lateral, we're going to have our fasciculus cuneatus that's carrying those same exact sensations except from the upper extremity. Now, coming off of our kind of posterior, our remnants of our posterior horn, we have our spinal trigeminal nucleus. And the spinal trigeminal nucleus is essentially just a continuation of the substantia gelatinosa of the spinal cord. And so if you remember from the spinal cord, pain and temperature fibers from the body would come through that posterior horn synapse in the substantia gelatinosa. So it actually makes sense that the spinal trigeminal nuclei are relaying that same pain and temperature information, except it's going to be those sensations from the head and face. Okay, so more lateral to the spinal trigeminal nucleus, we actually have a tract as well. And the reason that we have a tract that's actually visible in this section is that our trigeminal nerve fibers are entering and exiting at the level of the pons. But our spinal nucleus, remember, is extending all the way down to the inferior portion of the medulla. So fibers that are entering into the pons that need to get down to that nuclei, they have to descend until they get to their appropriate location within that nucleus. And so it's these descending fibers that forms that tract that's going to be lying just lateral to the nucleus itself that we can see on these digital sections. Now, lying anterior and somewhat lateral to the spinal trigeminal nuclei and tracts are going to be the spinocerebellar tracts that are carrying proprioceptive information from the extremities, and they're going to be ascending to the cerebellum. And then more anterior, we have our spinothalamic tracts, which are carrying pain, temperature, and crude touch sensations from the contralateral body. Okay, our second section is going to be at the level of the sensory or lemnisci decussation. So at this level, we can see our corticospinal fibers that are giving rise to the appearance of the pyramids in the most anterior portion of the medulla. And we can still see the spinal trigeminal nuclei and tracts in the lateral portion, as well as the spinocerebellar tracts that are lying just anterior to those spinal trigeminal nuclei and tracts. Now, a big difference that we see in this section is going to be the presence of these four large nuclei in the posterior portion of the medulla. So in the previous section, this area was largely occupied by the fasciculus gracilis and cuneatus, those dorsal column tracts. Now these tracts are replaced by the nucleus gracilis, more medial, and the nucleus cuneatus, more lateral, which is where these dorsal column tracts are actually traveling to synapse. So there's also a small little nucleus that's projecting off the lateral side of the nucleus cuneatus, right there. And those small little nuclei are just referred to as the accessory or lateral cuneate nuclei. And so these nuclei are actually receiving some of the fibers from that cuneate tract and then projecting to the cerebellum. So going back to the nucleus gracilis and cuneatus, we have second order neuron fibers that are leaving these nuclei, and they're actually kind of wrapping around this portion of the medulla, traveling anterior and medial to decussate. And when they decussate and cross midline, they're going to end up in this region and ascend in this position as the medial lemnisci. So these fibers that are wrapping around anteriorly and medially are referred to as the internal arcuate fibers. So the internal arcuate fibers leave the nucleus gracilis and nucleus cuneatus, cross midline, and ascend as the medial lemniscus. So our next section is going to be at the level of the inferior olivary nuclei. So once again, we have our corticospinal tracts that are forming the pyramids in the most anterior portion of the medulla. And remember, these tracts are above the level of the pyramidal decussation, so they have not crossed midline yet to get to the contralateral side. This means that these tracts in this location are just descending motor fibers coming from the ipsilateral motor cortex. Now, more posterior and lateral, we have these very large, uniquely shaped inferior olivary nuclei that have this characteristic kind of tapeworm look to them, and it's very distinctive. So anytime that you see it in a section, you know that you're going to be at the level of the medulla. And they're so large that they're actually producing a bulge on the external surface that we can actually see, and that bulge is referred to as the olive. So we have our pyramids. Those bulges were formed by the underlying corticospinal fibers, and then lateral to the pyramids we have the olives. That's due to the inferior olivary nuclei. And the pyramids and the olives are separated from one another by a sulcus. It's called either the pre-olivary sulcus, sometimes it's called the anterolateral sulcus. And in this section we can still see those dorsal nuclei, so those nucleus gracilis and nucleus cuneatus, as well as the medial lemnisci. And then those fibers that were leaving the nucleus gracilis and nucleus cuneatus, kind of wrapping around the anterior medial portion and then decussating to get to the medial lemniscus. Those are our internal arcuate fibers. Now our central canal is expanding at this point as it's going to approach the fourth ventricle, and just anterior to the fourth ventricle is going to lie our hypoglossal nuclei, which are just more medial in position. And if we look at the external brainstem, we can see the hypoglossal nerve right here as it's exiting the brainstem, and it's exiting in this level of the medulla, in between the pyramids and the olives in that anterolateral sulcus. So in the digital section, that means that nerve fibers are leaving that nucleus and traveling in between the pyramids and the olives. That's actually just a remnant on both sides in this section of where that hypoglossal nerve was located. So all of the other cranial nerve fibers that we're going to discuss in the medulla are actually exiting the medulla posterior to the olive. The hypoglossal is the only one that's exiting anterior to the olive in this anterolateral sulcus. Now, just lateral to the hypoglossal nuclei lie the dorsal motor nuclei of the vagus. Now, the positioning of these two nuclei, the hypoglossal nuclei and the vagus, create a bulge into the floor of the fourth ventricle that can once again be seen in the gross specimen. Okay, so now we're looking into the floor of the fourth ventricle. We've removed the cerebellum. And so this line right here is just the striatum medullaris that's kind of separating the pons above from the medulla below. And so these triangular shaped bulges right here are our trigones. So more superior and medial, we have our hypoglossal trigone, and then more inferior and lateral, we're going to have our vagal trigone. These trigones just reflect those underlying hypoglossal and vagal nuclei. Now, our medial longitudinal fasciculi are going to lie anterior to where the hypoglossal nuclei were located, and they're actually going to remain in this position relatively throughout the entire brainstem, that is anterior to the fourth ventricle, and any kind of motor nuclei that are present in that region, and all the way down to the spinal cord, once again anterior to the central canal and central gray matter. Now, one tiny nucleus that's very difficult to identify on most sections is the nucleus ambiguus, which is actually embedded in the reticular formation, and it's going to be posterior to our inferior olivary nuclei in this section. Now, this nucleus ambiguus is just giving off motor fibers that's going to form the glossopharyngeal nerve, and it's going to give off some motor output that's going to join with the vagus nerve as well. And then lastly, we have our solitary nuclei and its associated tracts that are lying posterior and lateral to the vagal nuclei. Now, these nuclei are actually receiving taste fibers that were traveling within the facial, glossopharyngeal, and vagus nerves, and it's also carrying some cardiorespiratory information that are being carried by the glossopharyngeal and vagus nerves. So the last section that we're going to look at in the medulla is going to be at the level of the pontomedullary junction. So at this level, we can see our lovely pyramidal corticospinal tracts, once again in that most anterior portion of the medulla. And we can also see just the superior tip of our inferior olivary nuclei at this location as well. Now on the posterior lateral surface of the inferior olivary nuclei lies these tract of ascending and descending fibers that's referred to as the central tegmental tract. And it contains ascending fibers that were traveling from the solitary nuclei that we saw in the previous section, and they're going to be ascending to the thalamus. And it's going to have some descending fibers that are traveling from the level of the midbrain to these inferior olivary nuclei. Now, more medial to the inferior olivary nuclei are going to lie our medial lemnisci. And then more lateral to our inferior olivary nuclei are going to be our spinothalamic tracts as they're ascending to the thalamus. Now these large circular masses in the posterior lateral sides of the medulla are going to be our inferior cerebellar peduncles that are just connecting the medulla to the cerebellum. And posterior and medial to these inferior cerebellar peduncles lies our vestibular area, where our 4 vestibular nuclei are going to be located. And then lateral to these inferior cerebellar peduncles lies our cochlear area, where our 2 cochlear nuclei are going to be found. So next we're going to look at the pons, and we're going to use 2 major sections to help guide our study of the pons. So our first section that we're going to look at is going to be at the level of the facial colliculus. Now at this level, our central canal has expanded into our 4th ventricle. So our 4th ventricle in this section is just this large white space that we can see in this posterior portion, posterior to the pons itself. Now the pons can be divided into 2 major sections. So more posterior, it's referred to as the tegmentum, and then more anterior, it's referred to as the basilar pons. So within the basilar pons, we have our descending corticospinal tracts, and then we've got a ton of small little nuclei that are just referred to as pontine nuclei. And from those pontine nuclei, there's quite a few fibers that are originating and decussating and traveling in this fashion, in a horizontal fashion, and those fibers are referred to as transverse fibers. So these transverse fibers, once they decussate, they're actually going to travel to the cerebellum, into this large mass on either side. And those transverse fibers actually make up this mass, and that mass is the middle cerebellar peduncle. So that middle cerebellar peduncle is made up of transverse fibers that are traveling, wrapping around the lateral side of the pons, going to the cerebellum. Now, in the tegmentum, we have facial nuclei, and posterior and medial to those lie the abducent nuclei. Now, fibers leaving the facial nucleus actually takes a unique course by traveling posterior medial, and it's going to wrap around the abducent nucleus, and then it's going to travel anterior lateral before it exits the pons. Okay, and so these facial nerve fibers that are wrapping around the abducent nuclei, they give rise to this characteristic bulge that's going to bulge into the floor of the 4th ventricle, and that bulge is referred to as the facial colliculus, which we can see in the gross specimen right here and here. Now, fibers that are leaving the abducent nuclei, they actually don't take quite a unique course. They just travel straight anterior throughout the tegmentum and the basilar pons before they exit. So we can see our vestibular area made up of our vestibular nuclei that's going to be lying just lateral to where the facial colliculi are going to be. And then once again, we can see our medial longitudinal fasciculi, which are medial in location, just anterior to that fourth ventricle. Now we can still see the spinal trigeminal nuclei and tracts as we saw in the medulla that's lying lateral, in this case, to the facial nuclei. And then the most anterior and medial part of the tegmentum is going to be where our medial lemnisci are going to be located. And then lateral to those medial lemnisci are going to be our spinothalamic tracts. Sometimes it's called the spinal lemnisci. And then posterior to our medial lemnisci is going to be our central tegmental tracts, which are just carrying those ascending and descending fibers. Now, something new here that we haven't seen before is the trapezoid body. And the trapezoid body contains fibers that originated in the cochlear nuclei. And they're going to wrap around the anterior portion of the tegmentum, decussate, and then ascend as the lateral lemnisci. And so we have to look back at this section over here. This is at the level of the pontomedullary junction. Remember, these were our inferior cerebellar peduncles, and our cochlear nuclei were just lateral to those peduncles. And so from these cochlear nuclei, we have fibers that are traveling in this direction, and they're going to be decussating, crossing one another, decussating, and then traveling once they decussate as the lateral lemniscus. Once again, it's these decussating fibers that give rise to that trapezoid body. Okay, so our last section of the pons is going to be at the level of the trigeminal chief sensory nucleus. And you should just be aware that the chief sensory nucleus is sometimes called the principal sensory nucleus and sometimes called the main sensory nucleus. So all the same thing, just right now I'm going to refer to it as the chief sensory nucleus. So the trigeminal chief sensory nuclei are lying in the posterior lateral tegmentum, just under the floor of the fourth ventricle. Okay, and just medial to the chief sensory nuclei are going to lie the trigeminal motor nuclei. All right, so we have our chief sensory, more lateral, and then our motor is going to be more medial. Now, posterior to the chief nuclei are going to be the smaller mesencephalic nuclei, and they're going to have a tract that's associated with them as well. Now these mesencephalic nuclei originate in the level of the pons at this level, and they're just going to project upwards, and so we can actually see them at the level of the midbrain as well. Now finally, all of the lemnisci that we've talked about so far, the medial, spinal, trigeminal, and lateral lemnisci are all present here, and they have more of a horizontal layout, kind of at the junction of the basilar pons and the tegmentum. Now this horizontal layout of the lemnisci is actually going to change once we get up into the midbrain, and we'll see that in just a little bit. It's going to change in shape and become more C or L shaped, and it just has to do with the underlying characteristics of where those lemnisci are actually going to be projecting to. Alright, and now very last, we're going to look at the midbrain, and we're going to use two major sections to help guide our study. And so first, we're going to look at the midbrain at the level of the inferior colliculi. And so if you remember from the gross specimen, when you look at the posterior portion of the brainstem, and you peel back that cerebellum, these four little bulges right here, those are our colliculi, superior and inferior colliculi. And collectively, all four of those are just referred to as the corpora quadrigemina, and they're making up the tectum of the midbrain. And one thing to notice from this kind of gross view is notice how the trochlear nerve, cranial nerve number 4, is exiting the brainstem and traveling underneath the inferior colliculus. Okay, so the trochlear nerve is the only nerve that's exiting the brainstem from the posterior portion, and then it's just going to wrap around the lateral brainstem before it travels anterior to get to the superior oblique muscle. Okay, so in this section, the inferior colliculi are lying in the posterior portion of the midbrain. And in the very anterior portion, we have these large bulges that are referred to as the crus cerebri. And these crus cerebri contain descending corticospinal fibers, as well as some descending fibers that are traveling to pontine nuclei and some of the various cranial nerve nuclei as well. So the tegmentum is just going to be the space that's in between the colliculi and the crus cerebri. So that whole space right there is referred to as the tegmentum. We had a tegmentum that was extending down into the level of the pons as well. Now, if you have the tegmentum plus the crus cerebri, so this whole area right here, that whole portion is referred to as the cerebral peduncle. Okay, now the large area that's going to be posterior to the crus cerebri is going to be the substantia nigra, which has very important dopaminergic input to the basal ganglia. And then these large circular masses in the middle of the tegmentum are the superior cerebellar peduncles, which began in the cerebellum, and then they're decussating at this level as these fibers ascend. We can also see our lemnisci in this section as well. Now remember, in the level of the pons, our lemnisci had that more horizontal shape. And like I said, in the midbrain, they kind of change shape to a C or L shaped, and that just has to do with where they are actually projecting to. So remember, back here is our inferior colliculus, and this portion of our lemnisci seems to be projecting straight back to that inferior colliculus. And the only lemnisci that's projecting to that inferior colliculus is the lateral lemniscus, which is carrying those auditory fibers coming from the cochlear nuclei. So this whole region back here is going to be where our lateral lemnisci are going to be located as they're traveling to the inferior colliculi. Okay, we can see our cerebral aqueduct at this level, which is connecting the third and fourth ventricles to one another. And then that cerebral aqueduct is surrounded by a bunch of gray matter. This is referred to as the periaqueductal gray, and that's going to be very important in some of the pain reduction pathways. Now, in the very anterior and medial position of the periaqueductal gray is where our trochlear nuclei are going to be located. So very tiny nuclei. And from the trochlear nuclei, those trochlear nerve fibers are actually exiting, passing posterior and laterally around that periaqueductal gray before turning medial. And they're actually going to decussate to the opposite side. When they decussate to the opposite side, that's when they're exiting the brainstem, coming out underneath those inferior colliculi. Now, kind of sandwiched in between the trochlear nuclei and the decussating superior cerebellar peduncles is where our medial longitudinal fasciculus is going to be located. And remember that medial longitudinal fasciculus is roughly staying in the same position all the way throughout the brainstem and even down into the spinal cord. Now we can also see our central tegmental tracts in this section as well, just lateral to where that medial longitudinal fasciculus is at in this section. All right, there's our central tegmental tracts. Now our very last section that we're going to look at in the brainstem is going to be at the level of the superior colliculi of the midbrain. So the superior colliculi essentially just replace the position of the inferior colliculi that we saw in the previous section. So they're just going to be in this posterior portion of the midbrain, making up the tectum. Now the crus cerebri and the substantia nigra are actually just remaining unchanged in this section compared to the previous section that we just saw. Now posterior to the substantia nigra lie these large round red nuclei. Now many fibers that were within the decussating superior cerebellar peduncles are actually going to terminate here. Not all of the fibers, but many of them. Once they decussate, they're going to go to these red nuclei and end there. Now, once again, the lemnisci that we looked at are wrapping around the lateral tegmentum. Now, one key difference here is that the lateral lemnisci are not going to be present here since those fibers were the ones that terminated in the prior section at the level of the inferior colliculi. And now our oculomotor nerve nuclei here is essentially just replacing where the trochlear nuclei were at in the prior section. So once again, just in that anterior and medial portion of the periaqueductal gray. And one thing to note here is you can actually see fibers that are exiting the oculomotor nuclei and traveling anterior through the tegmentum, through the red nucleus, and even some through the substantia nigra before they exit in between the crus cerebri in this interpeduncular fossa. Now, the tiny Edinger-Westphal nuclei are going to be lying just medial to the oculomotor nuclei, and they're so close to the oculomotor nuclei that sometimes both of them combine are just referred to as the oculomotor complex. Now, the Edinger-Westphal nuclei are just supplying some preganglionic parasympathetic input to the eye.